Hello everyone, I welcome you all. And my name is Snehlata, and I'm a PhD student and a PMRF fellow at the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Cell, the Unit of Life. So till now we have studied about the endomembrane system, and in the endomembrane system we have basically studied about three major organelles, that was endoplasmic reticulum, Golgi bodies, and lysosomes. Today, we will study about the last organelle of the endomembrane system, that is vacuoles. So, if we talk about the vacuoles, vacuoles are the spherical structure that is bound by a single membrane. So, a vacuole looks something like this. Okay. Now, this single membrane of a vacuole is known as tonoplast. So, the single membrane of this vacuole is known as tonoplast, and this tonoplast has various functions. So this structure is known as the membrane is known as tonoplast, and it has various functions. The first function of the tonoplast is it separates the cell from the vacuole from rest of the cytoplasm, right? So if you have a cell, so it separate the vacuole from cytoplasm, right? So this is the first function of the tonoplast. The second function of the tonoplast is it is selectively permeable. Okay. So just like the plasma membrane, it is also selectively permeable, which means it only allows certain selective molecules to pass through it. And how these molecules pass through this tonoplast? Their materials or the molecules pass through tonoplast via active transport. So the materials pass through active transport. So what does active transport means? Active transport means that for transporting these materials across the tonoplast, energy is required. So it requires energy, right? So these are the functions of tonoplast. Now, the next thing is what are the constituents of a vacuole? So, what a vacuole comprises of? So, a vacuole majorly comprises of water. Apart from this, is comprises of sap and excretory materials or the waste materials. So, it comprises of excretory materials or waste materials. So, these are the constituents of a vacuole. Now, just like lysosomes, we saw that there are 4 types of lysosomes. Similarly, the vacuoles are also of different type. So the first type of vacuole is known as sap vacuole. Okay, so the first type is sap vacuole. The sap vacuole is present in plant cells. So the sap vacuole is present in plant cells, and the membrane of sap vacuole is tonoplast, like I said. So the membrane of a sap vacuole is known as tonoplast, and it consists of water and minerals. Okay, so it consists of water and minerals. Now, how these minerals are transported? So these minerals are transported across tonoplast via active transport, right? And for active transport, what kind of channel or what kind of protein is required? The protein that is used for the active transport is known as pump. So basically, pumps are used to transport these minerals across the tonoplast. And this sap vacuole is very large in size, and it comprises or it constitutes 90% volume of a plant cell. So a sap vacuole constitutes 90% volume of a plant cell. Okay. Now, the next type of vacuole is contractile vacuole. So as the name suggests, it has contractile, that is contraction. So this vacuole can change its shape. This vacuole can change its shape. Now, what is the function of a contractile vacuole? So for that, let's take an example of a unicellular organism, that is amoeba. So the amoeba is found in fresh water, right? So it has water around it. Now, this water, we know osmosis, right? So in the osmosis, the water moves from higher concentration to low concentration. So the water will move from outside to inside. So in this case, what happens? This contractile vacuoles, they take this water. So this is your large vacuole. Inside this contractile vacuoles are present. These contractile vacuoles, upon taking the water, increase in the size. Okay. And then they transfer this water to this large vacuole, and this large vacuole excrete out the water. So in this way, it prevents the bursting of this amoeba cell. Otherwise, due to the osmosis, the water and there will be extension on in the cell, and the amoeba will burst, right? So this prevent osmotic bursting. It prevent osmotic bursting, or you can say it cause osmoregulation. So it helps in osmoregulation. Okay. Now the example of the organisms in which contractile vacuoles are present are amoeba and euglena. So these are some examples in which contractile vacuoles are present, and both of these organisms are present in fresh water. And because of this reason, the contractile vacuole is important for these organisms. Now, the third type of vacuole is food vacuole. So the food vacuole, as the name suggests, it contains or stores food, right? So again, the example is amoeba. So in amoeba, we have studied about a process known as endocytosis, right? So in the case of endocytosis, if you have a food, solid food, and it has to be taken inside, then this process is known as endocytosis. And since it is a solid food, so the process further can be said as phagocytosis, right? It can be called as phagocytosis. So the amoeba take the food via this process of endocytosis, and it does so with the help of a food vacuole. So the example for organism containing food vacuole is amoeba. The next type of vacuole is gas vacuole. But gas vacuole is not actually a vacuole, it is a pseudo vacuole. Okay. It is pseudo vacuole. Why it is called a pseudo vacuole? Because it does not have a membrane. It does not have a membrane. And these vacuoles, or gas vacuole is present in, when we were studying prokaryotic cell, we saw that the gas vacuoles are present in bacteria. So these are present in bacteria, such as blue-green algae, purple sulfur bacteria, etc. Purple sulfur bacteria. Okay. So this food gas vacuoles are not actually true vacuoles. These are not true vacuoles, but these are pseudo vacuoles that are present in bacteria. So with this, we have studied about the endomembrane system. Now let's solve few questions before moving forward. So the first question is, the endomembrane system of a cell includes, so we have already studied that the endomembrane system comprises of 4 organelles, that is ER, Golgi bodies, lysosomes, and vacuoles. So D is correct answer. The second is, the outer membrane of a nucleus is in continuous with the ER, does not be a ribosome. So if you remember the structure, so you have a nucleus, and this is a continuous of the nuclear envelope, and this structure is known as ERs, right? So the first option is correct. However, the second option is it does not be a ribosome, which is not true because the ER is further of 2 types, rough endoplasmic reticulum and smooth endoplasmic reticulum. And the rough endoplasmic reticulum have ribosomes attached to it. So because of this reason, only option 1 is correct, right? Now, the third question is, what is true about both A and B in the diagram? So if you see, the A here is representing, this is a diagram where A is representing rough endoplasmic reticulum, right? And the B here, it does not have ribosome, right? So it is tubule shape. So it is another type of endoplasmic reticulum, that is smooth endoplasmic reticulum, right? So What is true about this? Both of these, the first option is they are involved in protein synthesis. No, the only rough endoplasmic reticulum is involved in protein synthesis, right? The second option is they synthesize steroidal hormones in animal cells. The steroidal hormones were synthesized by SCR. The third option is they are a part of an endomembranous system of a eukaryotic cell. And this is the correct answer because both RER and SCR are a type of endoplasmic reticulum, which is a part of an endomembranous system. And the fourth option is they are continuous with the inner membrane of the nucleus. No, the ER is in continuous with the outer membrane of the nucleus, right? So the correct answer will be C. Now, after studying the endomembrane system, next we will move to another organelle that is present in most of the eukaryotic cell, and it is known as mitochondria. Okay, so in mitochondria, first let's discuss about the structure of a mitochondria. So mitochondria is double membrane bound structure, right? It is double membrane bound. Okay, so the mitochondria will look something like this. It will have double membrane with certain modifications that we will see. Now, if we talk about the shape of mitochondria, so the shape of mitochondria, as you can see, it is cylindrical in shape. It is cylindrical, or it is also known as sausage shape. Sausage shape. Okay, so the mitochondria is of cylindrical or sausage shape. The third thing about the size. So the size of a mitochondria, so it has a length and a diameter. Okay, so the length of a mitochondria is 1 to 4 micrometer and the dia is 0.1 to 1 micrometer. So we can write the length is 1 to 4 micrometer and diameter of mitochondria is 0.1 to 1 micrometer. Apart from this, if we talk about the number, that what number or how many mitochondria are present in a particular cell, so this number vary. Okay, so it is not important that all cells consist of an equal number of mitochondria. So the number of mitochondria vary from cell to cell. It varies from cell to cell. And on what basis the number of a mitochondria is decided in a cell? It is based on the physiological activity of that cell. Okay, so it is based on the physiological activity of a cell. Okay, now what do you mean by physiological activity? How much activity, how much work a cell has to do, or how much energy requirement a cell has. Okay, so it is also based on the energy requirement of a cell. Based on energy requirement of a cell. For example, if we talk about muscle cells, so a muscle cells have always have to move, right? So these are responsible for the movements. So in this case, mitochondria are in large numbers. So in this case, in the case of muscle cells, a large number of mitochondria are present in a particular muscle cell, right? But if we talk about the RBC, so we know in RBC, no mitochondria is present, right? So based on the type of cells, the number of mitochondria vary. Apart from this, one important thing is we said that a cell can be visualized under a light microscope, right? But if you want to see a mitochondria, can we see it under a light microscope? The answer is no. Mitochondria. So how do we see a mitochondria? Mitochondria can be visualized by compound microscope. Okay, compound microscope, but they cannot be seen without staining because these are colorless. Okay, so they can be visualized under compound microscope, but by using stain. And the stain that is used to see a mitochondria is known as Janus stain. Okay, so mitochondria cannot be visualized without staining. If you want to see a mitochondria, you have to use a compound microscope and stain it with the Janus stain. Okay. Now moving to the next part. So now we'll study the structure of mitochondria in detail. Okay. So if you see here, so the mitochondria consist of two membrane. Okay. We can see it consists of two membrane. The first membrane is outer membrane. The first is outer membrane, and the second one is inner membrane. So that's why it is known as double membrane bound organelle, right? So it has an outer membrane and an inner membrane. And in between the outer and inner membrane, there is a space, and this space is known as intermembranous space. This space is known as intermembranous space or perimitochondrial space. It is also called as perimitochondrial space. Okay. So apart from this, what we can see that on the outer membrane there is this structure, this pore-like structure, and this structure is known as porin. Okay, so it is made up of special protein that is known as porin. Apart from porin, we can see that there is in the inner membrane there is this structure, and this structure is called as cristae. This structure is called as cristae. On this cristae, there is this stalk-like structure, and this is known as F0 F1 synthetase. So this is a particular enzyme. This is an enzyme. This is an enzyme then known as F0 F1 synthetase for oxysome. Okay. Now, apart from this, what else we have? We can see this is the green circles, right? So these are the enzymes. These are other enzymes that are present, and these enzymes are for ETS or electron transport chain. These enzymes are for electron transport chain. So electron transport chain basically comprises of a series of enzymes that are responsible for the process known as oxidative phosphorylation. For now, you just remember that these are the enzymes that comprises of electron transport chain or ETS chain that is responsible for oxidative phosphorylation. Okay. Apart from this, what else we have? We can see that there is this inner lumen, and this is known as matrix. This is known as matrix of the mitochondria. So in a mitochondrial structure, these are the components that are present. Now, we will discuss each of these components one by one. So if we talk about first thing, that is outer membrane. So we talk about outer membrane, what we saw that it is also, you can see the outer membrane itself is also double layer, right? So the outer membrane is double layer, and it is made up of lipid, just like a plasma membrane. The outer membrane of mitochondria is also made up of lipid. Apart from this, what else we saw? We saw that porins are present on outer membrane. So on outer membrane structure, porins are present, and because of this porins, it makes the outer membrane more permeable. Okay. Then there is no cristae on outer membrane. So cristae are absent on outer membrane, and also there are few enzymes. So there are less, or you can say there are few enzymes present on outer membrane. So this is all about outer membrane. Now the second was inner membrane. The second is inner membrane. So what we saw in inner membrane, again, this inner membrane is also double membrane bound, right? So it is also double layer. So the inner membrane is also double layer, and it is also made up of lipid. It is also made up of lipid, right? Apart from this, does the inner membrane consist of porin? No. So porins are absent. Porins are absent in the inner membrane. Thirdly, this inner membrane is less permeable. So it is less permeable. It allows only selective molecules to pass through it. It is less permeable. It has cristae. So the inner membrane comprises of cristae, and it also comprises of enzymes. So enzymes are attached on the inner membrane, and the enzymes are for ETS, that is electron transport chain, and we also saw one more enzyme, that was F0 F1 synthetase. So these are the enzymes that are present on the inner membrane. Now, the third component that we saw, the third component was cristae, right? So what was cristae? It was infolding of inner membrane. Infolding of inner membrane, right? So the cristae is infolding of inner membrane, and it was a cristae on which this F0 F1 in synthetase and the enzymes for ETS are present, right? So on the inner cristae is part of the inner membrane, right? On this cristae, the enzymes for ETS and F0 F1 synthetase are present, right? Apart from this, what is the function of cristae? So it is a site for oxidation and reduction. So what do we mean by site for oxidation and reduction? Like I said, that it comprises of these enzymes, right? And these enzymes are part of electron transport chain. So these enzymes undergo continuous oxidation and reduction, and hence leads to the formation of ATP. So, and this process takes place at cristae. Hence, it is known as site for oxidation, and reduction. Okay, it is called a site for oxidation and reduction. Secondly, this cristae, because of this infolding, it increases the surface area. So the cristae, it increases the surface area. Okay, so these two are the function of cristae, that they are site for oxidation and reduction, and they also help in increasing the surface area. Now, the fourth thing that we saw was intermembranous space. So intermembranous space. So what is intermembranous space? We just saw that it is space between intermembranous space between inner and outer membrane, right? So it is a space between inner and outer membrane, and this intermembranous space has low pH. Okay, so we have one chapter of oxidative phosphorylation where you will see that how ATP production and how ETS work. But for now, you just remember that the intermembranous space have low pH because it has low pH because it collects H plus ion. Okay, so it has lot of H plus ions. Okay, and because of this hydrogen ions, the intermembranous space have low pH, or you can say acidic pH. It have acidic pH. Okay. Now the fifth component of the mitochondria is matrix. So the fifth component of mitochondria is matrix. Where the matrix is present, it is present inside or inner to inner mitochondrial membrane, right? Present inner to inner mitochondrial membrane. Where it is present right here. This is the matrix part. This part is matrix, and it is present inner to inner mitochondrial membrane. Now, what does a matrix comprises of? So a mitochondria have its own DNA, okay, and this DNA is present inside the matrix, and this DNA is single. It is single, double-stranded. It is single, double-stranded, circular DNA. Okay. So mitochondria have its own DNA that is single, double-stranded, and circular. And this DNA, so it will look something like this. So it is double-stranded, okay, and circular. So it is double-stranded circular DNA, and it is present in the matrix. Apart from DNA, the matrix of mitochondria also comprises of ribosomes. So ribosomes, apart from cytoplasm, they are also present in mitochondria. Then this DNA can be converted into RNA, right, through the process of transcription. So the matrix also consists of RNA. And apart from this, it also comprises of certain enzymes that are crucial for some processes, such as Krebs cycle. So these have enzymes for Krebs cycle, or you can say TCA cycle. Apart from this, it also have enzymes for fat oxidation. So it have also an enzyme for fat oxidation, that is breaking of fat for the production of energy. And then enzymes for amino acid synthesis. It also have enzymes for amino acid synthesis. Okay, so this is all comprises of matrix. Now, one more important thing. So if you see the structure of mitochondria, you can see that there are two compartments, right? There is one inner compartment and outer compartment. So you can write that mitochondria have two compartments. Mitochondria have two compartments, right? You can see this is one compartment and this inner is one compartment. So it has one inner compartment that comprises of matrix. So inner compartment and the outer compartment. And what outer compartment comprises of? It comprises of intermembranous space, right? So the mitochondria have two compartments, the inner compartment and the outer compartment. Okay. So now we have seen the structure of the mitochondria and we have studied about each of these structures in detail. Now let's see what is the function of a mitochondria. So the major function of a mitochondria is, so the major first most important function of mitochondria is oxidative phosphorylation. The major important function is oxidative phosphorylation. Okay. Now what is oxidative phosphorylation? It is a process of formation of ATP. So oxidative phosphorylation is process of formation of or synthesis of ATP. Okay. Now, what is ATP? One second. Sorry. So, okay. So here we'll write this. The function. The first major function is oxidative phosphorylation. The first major function is oxidative phosphorylation, which is process of synthesis of ATP. Process of synthesis of ATP. And this ATP is known as energy currency. Right? It is known as energy currency of cell. So a cell have energy in the form of ATP, and ATP is known as energy currency of cell. Now, how does the mitochondria perform this oxidative phosphorylation? So we saw this structure, right? This F0 F1 synthase structure. So let's draw this structure. So it has a base. If we zoom this structure, we'll see it has a base, and then it has a structure something like this. So this structure, this is known as stalk. Okay, and this is known as head. And the head of this enzyme comprises of F0 F1 synthetase. So as the name suggests, it consists of a word synthetase that comes from word synthesis, right? So this F0 F1 synthetase is responsible for ATP synthesis. This is responsible for ATP synthesis. And from what it synthesizes the ATP? It requires ADP plus phosphate. So with the help of ADP plus phosphate, ATP synthesis is done. And this is done with the help of an enzyme known as F0 F1 synthetase. So the major function of mitochondria is oxidative phosphorylation. Apart from this, the second function of mitochondria is muscle contraction. Okay, so it also helps in muscle contraction. Thirdly, it helps in nerve signaling. So the signal transfer or passage of signal from one body part to another. So it also helps in this. Fourth, we saw that it also contains enzyme for fat oxidation, right? So fat oxidation or breaking of fat for the production of ATP is also done in mitochondria. The fifth function is it also involved in the synthesis of some amino acids. So it also helps in some amino acid synthesis. What are amino acids? These are the building blocks of protein, right? So the protein is synthesized with the help of the amino acids, or they are the chain of amino acids. And some of these amino acids are synthesized in the mitochondria. And because mitochondria has its own ribosome, so protein synthesis also takes place in mitochondria, right? So certain protein synthesis also takes place in mitochondria. So these are the major function of mitochondria, right? Okay. Now, next, now we have studied about the mitochondria. The next thing that we'll study is another double membrane bound organelle, and this double membrane bound organelle is known as plastid. Okay. So what is plastid? So we can write the plastid. If you talk about the structure of a plastid, so a plastid is again a double membrane bound structure, right? So it is double membrane bound. Okay. So plastid will also look something like this. It has two membrane. Now, if we talk about the shape of a plastid, so the shape of a plastid varies. Okay, so it can be either lens shape that is present in majority of plants. So it is in lens shape. Apart from this, the plastid can also be of ribbon shape. Okay, it can also be of cup shape. So this cup-shaped plasmid is present in an organism known as Chlamydomonas. Okay, so the cup shape is present in Chlamydomonas. Now, third thing, if we talk about the size of a plastid, so these are larger than mitochondria. Okay, they are larger than mitochondria, although this is also double membrane bound organelle. It is larger than mitochondria, and it has a diameter of 1 to 4 micrometer, but the length is of 5 to 10 micrometer. So it has length of 5 to 10 micrometer and diameter of 1 to 4 micrometer. Now, if we talk about its presence, so it is present in some protists. Okay, so protista is a kingdom. So it is present in some protista like diatoms, dinoflagellates, and euglenoids. Okay, so these are present in some protists. Apart from this, these are present in plants. Okay, and in plants, they are present in all plant cells. All plant cells. So plastids are present in all type of plant cells. One important point is plastids are absent in bacteria. Okay, so we'll see later that the major function of plastid is photosynthesis, and bacteria also do photosynthesis. But these plastids are absent in bacteria. But instead of plastids, the bacteria contain special enfolding of membrane, and that enfolding was called as that enfolding of membrane is called as chromatophore. So instead of Instead of plastids, the bacteria consist of chromatophores that help in photosynthesis. And the second thing is, unlike mitochondria that requires stain to visualize or so that it can be seen under a microscope, the plastids, they do not require any stain. So they can be easily seen in compound microscope without staining. Okay? Microscope without staining. So they do not require any staining to be seen in the microscope because they have their own pigments. So they have pigments, because of that they can be seen without stain. Okay. Now, if we talk about the types of plasmids, the plastids, so there are basically three types of plastids. So the plastids are of three types based on the pigments they have. So it is based on pigments they have. So the first type of plastid is known as leucoplast. So the first type is known as leucoplast. And the leucoplasts are colorless. So they do not have any color and hence they do not have any pigment because pigment is responsible for color, right? So the leucoplasts do not have any pigment and hence they are colorless. Apart from this, they do not have grana. So grana is a special structure that we will see in the next slide. But for now, just remember that grana is absent in leucoplast. Okay. Now, the leucoplasts are of three types. So the leucoplasts are of three types based on what they are storing. Okay. So based on storage, leucoplasts are of three types. The first type is helioplast. And the helioplast, what it stores fat and oils. Okay. So the helioplast stores fats and oils. The second type of leucoplast is amyloplast. The second type is amyloplast, which stores starch. And the third type is aleuroplast. The third type is aleuroplast that stores protein. So based on the type, what the leucoplast is storing, it is of three types: helioplast, amyloplast, and aleuroplast. Now, where the leucoplasts are present? So the leucoplasts are present in stems. They are present in stems and roots majorly. And they are also present in those fruits or plants that are found underground or that grow underground, such as potato. Okay, so the leucoplasts are also present in potato. So this is what we studied about the leucoplast, that is a type of plastid. Now the second type of plastid is chromoplast. Second type is chromoplast. Now, what is chromoplast? As the name suggests, it has word chroma, which means color. So this consists of, chromoplast consists of color pigments. These consist of color pigments. And what these color pigments are called as carotenoids. These are called as carotenoids. Okay. So the color pigments that is present in a chromoplast is known as carotenoids. And these carotenoids gives different colors such as red, yellow, and orange. So the carotenoids, red, yellow, and orange. Okay. So the carotenoid gives red, yellow, and orange color. And the chromoblasts are present in what region or which region of a plant? So they are mainly present in the aerial part of plant. So the plant that is above the part of a plant that is above the ground. So these are present in aerial part of plant. Aerial part of plant, such as fruits. Okay, so we can see different color of fruits, right? Fruits, flowers. So we can see different colors of these fruits and flowers. This is because of the chromoplast. Now, the third type of plastid is chloroplast. So chloroplast. So the chloroplast is derived from the word chloro, that comes from chlorophyll. So these are chlorophyll containing. So chlorophyll is another pigment. So these are chlorophyll containing plastids. Okay. Apart from chlorophylls, they also comprises of carotenoids. So the chloroplast comprises of two pigments. They comprises of two pigments, that is chlorophyll and carotenoids. And because of this chlorophyll, the chloroplasts are green in color. Okay. So the green color is because of chlorophyll. So chloroplasts are green in color. Now, where these chloroplasts are present? So they are present in all cells, but they are present majorly in mesophyll cells. They are present majorly in mesophyll cells of plants, plant leaves. Okay. So they are present majorly in the mesophyll cells of plant leaves. And the number of these chloroplasts also vary from cell to cell. For example, if you talk about mesophyll cells, so in mesophyll cells, these number is from 20 to 40. So in a single mesophyll cell, 20 to 40 chloroplasts are present. Okay. But if you talk about another organism, that is chlamydomonas, that is an algae. So in this, eight chloroplasts per cell are present. So 8 chloroplasts are present. Also, the shape of this chloroplast vary. So the shape of chloroplast can be different. So they vary, and they can be discoid shape, oval shape, star-like. Okay. So based on the type of cell, the size of the shape of chloroplast and their number also varies. So we have seen the three different types of plastid, that is leucoplast, chromoplast, and chloroplast. Now we'll see in detail about chloroplast. So if we talk about the structure of a chloroplast, we can see just like mitochondria, it also has two membranes. The first outer membrane, and the second inner membrane, right? And then they also have intermembranous space that is between outer and inner membrane. Apart from this, what else we can see? We can see that they have this stack-like organization, right? And one unit of this stack is known as grana. So the one unit of this stack is known as grana, and the complete stack, if you talk about the complete stack, this is known as thylakoid. So if you talk about the grana, it is a single entity of this entire stack. And if we see, this grana is double layer, right? So you can write grana is double layer, and it consists of certain enzymes. What enzymes? We'll see in next slide, but it comprises of certain enzymes. Similarly, if you see thylakoid, this thylakoid is stack of grana, right? It is stack of grana. And apart from this, we can see that these thylakoids are attached to each other, or they are connected to each other via a structure that is known as stromal lamellae. So this structure is known as stromal lamellae, right? Then this chloroplast also have inner matrix, right? This is the matrix. So the chloroplast also have matrix. This has also a matrix. Now, what are the constituents of the matrix of chloroplast? So like like mitochondria, the chloroplast also consists of double-stranded circular DNA. So the chloroplast also have its own DNA. Apart from this, it has ribosome, that is 70S. Mitochondria also have 70S ribosome, and it also comprises of certain enzymes. Okay, and the matrix of this chloroplast is known as stroma. Okay, so the matrix of chloroplast is known as stroma. Stroma is the matrix of chloroplast. Okay, so this is what we saw about the structure of the chloroplast. Next, we will talk about the function of the chloroplast. So the major function of the chloroplast is photosynthesis. Okay, the major function of chloroplast is photosynthesis. But when we'll study about the photosynthesis chapter, we'll study in detail. But for now, you should know that photosynthesis also comprises of two parts of the reaction. The first is known as light reaction. Okay. Now, if we talk about the light reaction, where it is performed in grana. Okay. So the light reaction is performed here. So it is performed in the grana. Why? Because grana has pigment that can perform this light reaction. Okay. Now, light reaction leads to the formation of ATP. So it leads to the formation of ATP and NADPH2. So this NADPH2 also upon further breakage leads to the formation of ATP. Okay. So if someone asks that where the ATP formation takes place in the chloroplast, it takes place in the grana. Okay. Now, if you remember, in the mitochondria also ATP formation was done, and that process was known as oxidative phosphorylation, right? But in the case of chloroplast, the formation of ATP, because it is taking in the presence of the light, it is known as photophosphorylation. So it is known as photophosphorylation. Okay. So light reaction is performed in the grana, in which the formation of ATP takes place, and this process is known as photophosphorylation. Now, the second part of photosynthesis is dark reaction. Okay. So second part is dark reaction. And where does this dark reaction takes place? It takes place in the stroma. So what is stroma? Stroma is the matrix of the chloroplast, and the dark reaction takes place in stroma because stroma has a special enzyme that catalyzes this reaction, and this enzyme is called as rubisco. Right? And this leads to the formation of or synthesis of sugar. So the synthesis of sugar takes place in the stroma, and the synthesis of formation of ATP takes place in grana. Apart from this, the second function of chloroplast is storage of starch. So this is the function of chloroplast. So we studied about mitochondria and chloroplast, and both of these are double membrane bound organelles. Now, if you see the lecture and if you remember, you can see that there are certain similarities between mitochondria and chloroplast, right? For example, both of them have single double-stranded circular DNA, right? So both mitochondria and chloroplast have double-stranded circular DNA. And these both mitochondria and chloroplast are known as semi-autonomous organelles. Okay. Now, what do you mean by semi-autonomous organelles? That can partially perform their own functions. Okay. So semi-autonomous means can partially perform their function. Okay. So they do not need to be dependent on the nucleus entirely, because nucleus contains genetic material, right? And that governs what a cell has to do. But this mitochondria and chloroplast, they do not rely entirely onto or depend entirely onto the nucleus. they can by themselves perform their function partially. Okay, so they can partially perform their functions. Okay, and because of this, they are known as semi-autonomous organelle. Now, if someone asks that how can you say it is a both are semi-autonomous organelle, you can write because they both have their own DNA, right? So they have own DNA that is double-stranded, that is double-stranded circular, right? So they have double-stranded circular DNA. Apart from this, they have their own ribosome, right? They have their own ribosomes, right? And because they have ribosomes, they can make their own protein. They can make their own proteins. And because of this, they are known as semi-autonomous organelles. Okay. Apart from this, they are also called as both are known as self-replicating organelles. They are known as self-replicating organelles because they have their own DNA and they divide by fission. Okay, so they have their own DNA which replicate by itself and they divide by fission. So fission is a process in which there is one cell and this cell then breaks or divide into two cells. So they divide by fission. Okay. Now, apart from this, it is said that both mitochondria and chloroplasts are evolved from bacteria, and this theory is known as endosymbiotic theory. So, what is endosymbiotic theory? Endosymbiotic theory says that both mitochondria and chloroplast evolved from bacteria. Now, if someone asks that what is the proof of this, so you can see that both have this double-stranded circular DNA, right? And the double-stranded circular DNA is also present in bacteria, right? So because of this double-stranded circular DNA, secondly, they both have 70S ribosome. They have 70S ribosome. A typical eukaryotic cell consists of 80S ribosome, but these two have 70S ribosome that is present in a bacteria. Thirdly, we know that DNA comprises of ATGC. These are the nucleotides. And the bacterial genome is known to have GC rich, known to be GC rich. Similarly, this circular DNA of mitochondria and chloroplast, these are also GC rich. It means they have high content of G and C, that is guanine and cytosine. Okay. And lastly, both divide by fission, like bacteria. So both divide by fission. And based on this, you can say that, yeah, both mitochondria and chloroplast have evolved from bacteria. Okay. So this is what we saw the similarity between mitochondria and chloroplast. Now the next thing that we will study is ribosomes. So what are ribosomes? These are also organelles, but they do not have any membrane. So do not have membrane, right? So our ribosomes do not have membrane or membraneless organelles. Secondly, these are smallest organelle. Thirdly, these organelles are ribosome is the only organelle that is present in both prokaryotes and eukaryote. And because of this reason, it is known as universal organelle. It is known as universal organelle. Why? Because present in both, it is a single organelle that is present in both prokaryotes and eukaryote. Okay. So because of this reason, it is known as universal organelle. Apart from this, if you talk about the location of the ribosome, so we we just saw that ribosome is present in mitochondria, right? It is present in mitochondria. It is present in chloroplast. We saw, right? Apart from this, we also saw when we were studying endomembrane system, it is present on rough endoplasmic reticulum. So it is also present on rough endoplasmic reticulum. And apart from this, it is also present in cytoplasm. Okay, it is also present in cytoplasm. And they are also present in prokaryotic cells. Prokaryotic cells, cytoplasm, right? So in the eukaryotes, these are present in mitochondria, chloroplast, rough endoplasmic reticulum, and cytoplasm. And in prokaryotes, these are present in prokaryotic cell, cytoplasm. Okay. Now, if we talk about the discovery, so the ribosome discovery, if we talk about discovery, so the ribosome discovery took place in 1953. So in 1953, there was one scientist named Palade. So in 1953, a scientist named Palade, he observed one structure in the cytoplasm that was granular in nature. So observed granular structure in cytoplasm. Observed granular structure in cytoplasm, which he called as ribosome. Okay. Now, if we talk about the structure of a ribosome, so if we talk about the structure, so ribosomes have two subunits. Ribosome have two subunits. Okay. So one is large subunit and one is small subunit. So larger subunit and smaller subunit. Okay. Smaller subunit. Now, the size of if we talk about the size of ribosome is measured in a special unit. Size means sedimentation coefficient. Sedimentation coefficient. So the size of ribosome is measured in unit known as sedimentation coefficient, or it is also known as Swedberg. It is also known as Swedberg. Okay. If we talk about the composition of ribosome, so it comprises of RNA plus protein. So the ribosome comprises of RNA plus protein. Now there are two types of ribosome. Okay, based on their size. The first is ATS. Okay, the ATS is present in eukaryotes, cytoplasm and areas. Okay, so eukaryotic cell, cytoplasm and rough endoplasmic reticulum consist of ATS ribosome. This ATS, if you draw the structure, it will look something like this. So it is a smaller unit and a larger unit. The smaller unit is of 40S and the larger unit is 60S. So these are not additive. It doesn't mean that the total should be ATS. It is a type of a unit, okay, based on the sedimentation coefficient. So the smaller unit is of 40S and the larger is of 60S. Now the smaller, if you break the smaller further, the smaller comprises of one more subunit, that is 18S. Okay, so it comprises of 18S, and the 60S further comprises of three subunits, that is 28S, 5S, and 5.5S. Okay, so this larger subunit comprises of 60S, which further, if we break down, comprises of three subunits, 28S, 5S, and 5.5S. And these are present in the eukaryotic cytoplasm and rough endoplasmic reticulum. Now, the second type of ribosome is 70S. Again, these are present in prokaryotes. These are present in prokaryotes and in mitochondria and chloroplast. In mitochondria and chloroplast. And if we break this, So it further comprises of two subunits. The one is 30s and the second larger is 50s. Now the 30s further it comprises of 16s and the 50s, if you break, it comprises of 23s subunit, 23s subunit and 5.8s subunit. Okay, so these are the types of ribosomes based on their size, and these subunits comprises of two subunits which further can be broken into more subunits, right? So this is all about the ribosome. In the next lecture, we will see about the cytoskeleton and nucleus. That's all for now. Thank you.